micro, okay, microscopic particles, microscopic organisms. So they can be seen only with the help of a microscope. They are called, they are called microorganisms. Microorganisms. One more definition also, an organism that can be seen only with the help of a microscope, and that typically consists of only microscopic single particles, microscopic cell. Organisms. Okay, so they can be seen only with the help of a some of the microorganisms are shown in figure 2 to 6. Let us observe the different types of microorganisms. Okay, very important microorganisms. See now, coccus. This is a coccus. Please observe carefully. Your spelling will be different and pronunciation will be different. So you have to find out that and spelling also you have to learn. Your different types of spellings are there here. Coccus, coccus. Next, second one is bacillus. Okay, bacillus. Next, lactobacillus. Lactobacillus. This is a figure number 2. Different shapes of bacteria. Coccus, bacillus, and lactobacillus. These are the different shapes of bacteria. Different shapes of bacteria. Let us go for that one more microorganism, that is fungi. Okay. So here, penicillium. Okay, what is that? Penicillium. Next, rhizopus. This is also called bread mold. Bread mold. Next, aspergillus. Next one is aspergillus. These are the fungi, comes under fungi category. Next, let us go for that figure number 4, protozoa. Protozoa. In this protozoa, you are very familiar with this amoeba, which does not have a definite structure. Okay, shape. Next, paramecium, not paramoecium, here paramecium. Okay, paramecium like a paranchepal. This structure will be like our paranchepal slippers. Okay, so here next, verticella. Verticella. Okay, let us recall once again the microorganisms which are given here. Observe carefully. Figure number 2, different shapes of bacteria. Different shapes of bacteria. Here, coccus, first one. Second one, bacillus. And third one, lactobacillus. Lactobacillus. Next, what about fungi? Figure number 3, fungi. Penicillium. Next, rhizopus, bread mold. Next, aspergillus. Aspergillus. Next, figure number 4, protozoa. Protozoa. Here, amoeba. Amoeba, unicellular organism. Single cell. It consists of single cell. Next, paramecium. Think about paramecium. Paramecium. Next, verticella. Verticella. These are the some of examples for bacteria, fungi, and protozoa. Okay. Next, here figure number 5. This is about algae. What is that? Algae. So here we can see a beautiful structure figures here from algae. Chlamydomonas. See children, here very important. These are structures. Amoeba structure. They may ask to draw. And next, bread mold structure, paramecium. All are important, but in that mostly they can ask to draw in exam. Please observe carefully the structures. Next, coming to algae, chlamydomonas is also very important to draw the diagrams. Okay, so here chlamydomonas. Chlamydomonas. They all are comes under that algae. Next, Spirogyra. Spirogyra. Next, C, diatom. What is that? Diatom. Next, D is spirulina. Spirulina. Next, here, E, Idogonium, not Oidogonium. It is Idogonium. Please observe carefully, all of you. Figure number 5, E, Idogonium. Idogonium. Oedogonium. Idogonium. Next, figure number F, Serratium. This is Serratium. These all are comes under algae. Next, let us go for figure number 6, microarthropods. Microarthropods. In this, Cyclops. What is that? A Cyclops. Next, B is Daphnia. What is that B? Daphnia. Next, C is Scabies mite. Scabies mite. These are the like responsible for that allergy, itchiness. Okay. Scabies mite. Scabies mice. Next, eyelash mite. Eyelash mite. Okay, not neelash that. Okay, that is eyelash mite. Okay, fine. Let us recall once again the figure number 5, figure number 6, algae and microarthropods. Now, figure number 5, algae. A, Chlamydomonas. What is that? Chlamydomonas. Next, B is Spirogyra. B is Spirogyra. Next, C is Diatom. Diatom. Next, D, Spirulina. E, Idogonium. Oedogonium. Idogonium. And last one, F, Serratium. What is that? Serratium. Okay. Let us go for the microarthropods. The first one, for the first arthropod, A is Cyclops. Okay, children? Cyclops. Next, B. Look at the screen, observe that figure. B is Daphnia. Okay. Next, C is Scabies mite. Scabies mite. Next, D is eyelash mite. D is eyelash mite. These all are the these all are microarthropods. Arthropods. Now, let us study about some of a group of organisms. Group of organisms. Okay. Group of organisms. Here, let us study some microorganisms that belong to the groups like bacteria, fungi, protozoa, algae, and certain microarthropods with the help of some activities. Let us study some microorganisms that belongs to the groups like bacteria, fungi, protozoa, algae, certain microarthropods with the help of some activities. Already we have observed some of examples for this. For this, we need a microscope. You already know how to use it, and you could also refer to chapter Cell, the basic unit of life. Okay, how to use the microscope? You have to refer for the cell, the basic unit of life. Okay, now let us go for that activity to observe the different microorganisms like bacteria, fungi, and algae, and microarthropods, etc. See here, microorganisms in water. Very interesting, children. Microorganisms also present in water. So to observe that microorganisms in water, we need to collect some pond water or water, normal water, tap water from any tank in your surroundings. Collect. Why we need to take this pond water or tank water? Why we should not take mineral water? Because mineral water is already mineralized. We may not get microorganisms in that water. So if we collect pond water or from tank, there is have chance to observe some of microorganisms in that water. So we need to collect some pond water or water from any tank in your surroundings, take some of the greenish scrapings from the side of the tank. Okay, the side of this tank, you will observe that greenish scrapings already. So you have to take some of the greenish scrapings from the side of the tank and take 1 to 2 drops of water from the sample you have collected. Okay, you have to take the greenish scrapings and what water you have collected from the tank or pond water. You have to collect take 1 or 2 drops of water from that collected sample. Where we have to take that on a slide. You just with the help of dropper, you put 1 or 2 drops of water on a slide and observe it under the microscope. Simple technique. There is no need of keep one more slide, and there is no need of here cover slip or any other things. Simply, if you take if you collect water from pond or tank, from that with the help of dropper, you have to take 1 or 2 drops. You just put 1 or 2 drops of water on a slide and observe under the microscope. Draw rough sketches in your notebook of what you have observed. It is necessary. Don't forget to draw that observation diagram in your observation book. While you are observing, you need to take a observation book with you, and while you are observing, you need to draw the diagram of your observation. It is must. Okay, don't forget to draw that. Okay, so draw rough sketches in your notebook of what you have observed. Compare it with, okay, compare it with the figures given above for observation and identification. You may also hold a discussion about their shape, size, and other characteristics with your friends. Okay, take your help, teacher help also, with help of teacher, and you just form as like groups in your friends, and you just observe that, and next you may also hold a discussion about their shape, size, and other characteristics with your friends and teacher. Okay, take your teacher help also. And can you name the organisms which you have observed through the microscope? Already you have here a rough sketch diagrams. Here we have many of the organisms or diagrams in your textbook. You just compare that rough sketch diagram, what you have observed, the observation diagram, with your textual diagrams, and expect and name the organisms which you have observed through the microscope. We will try to know more about the microscopic world by doing the following activities. By doing the following activities, we can know the microscopic world. Okay. To get more information, let us go for the following activities. And before that, we have to know about fungi also, because fungi also one of the microorganism. Microorganism. Okay. Here, now let us know about the fungi. Okay, I will tell you one definition about fungi. A fungi, a single-celled or multi-celled organisms. Okay, fungi maybe single-celled or multi-celled organisms. Okay, single-celled or multi-celled organisms without chlorophyll. Without the chlorophyll, that reproduce by spores and lives by absorbing nutrients from organic matter. Fungi, nothing but a single celled or multi-cellular organism. Okay, without chlorophyll, that does not consist chlorophyll to make their own food like as plant, and that reproduce by spores. Okay, this is very important, that reproduce by spores and lives by absorbing nutrients. How they can live? By absorbing nutrients from organic matter. They depend on organic matter to absorb the nutrients. That is a fungi. Okay, fine. Then let us observe what is there in our textbook about the fungi. So usually after the rainy season, you might have seen some small umbrella like grows over rotten materials. Okay, spoiled materials. Please stand line. Okay. Usually after the rain season, you might have seen some umbrella-like grows over rotten materials or dumped waste. Yes, you have experience with this. You have seen some umbrella-like grows over rotten materials of dumped waste. Okay, dumped waste. Did you think about that dumped waste, like which is grow on that particular dumped waste materials? Rotten materials like umbrella-like structures. So here, between the grasses in a field and edges of wet rotten wooden planks also. Between the grasses in a field and edges of wet rotten wooden planks. You can observe. Often you might have observed white patches on the bark of trees. You might have observed white patches on the bark of trees. These patches are formed due to fungi. The white patches, what you have observed, the white patches, what you have observed on the bark. Bark means what? Todka. Bark of trees. These patches are formed due to the fungi. Now let us look at them more closely by the following activity. Here we have a great activity, very easy activity you can also do at home. Okay. Now activity 2. So here the activity is photograph of a card in bowl. Photograph of card in bowl. Very simple activity and interesting activity. So let us know about that to observe that particular microorganisms. Take some rotten part of vegetable or a black spoiled part of a bread or coconut with the help of a needle. Don't touch with hand because in your hand also maybe thousands of microorganisms are present. So while observing under microscope, we may get confused and we may not get a proper image. That's why you need to use a particular that needle to collect. To collect that, you need to use that needle. So take some rotten part of vegetable or black spoiled part of bread or coconut with the help of a needle. Okay, with the help of a needle, you have to take that. Okay, next. With help of a needle, after collecting that sample, you place it on a slide. A particular black mark, you place it on a slide and put a drop of water. Put a drop of water with a dropper. Okay. After that, you have to place a cover slip on it gently. Okay. You have to collect with the needle that particular black spots and put on that slide. So on that sample, you have to take a drop of water, place a cover slip on that particular sample gently and observe that sample under the microscope because these are the microorganisms which can be seen under microscope only. That's why we need to use the microscope to observe that particular microorganisms. So here this is figure number 7, photograph of card in bowl. So let us go for that continuation here. Next, draw rough sketches in your notebook of what you observed. Take the help of the figures given in figure 3, bread mold, rhizopus, and compare. Okay, here we have rhizopus. Here we have rhizopus. Where it is? Rhizopus bread mold. See. So here we have rhizopus bread mold. So you just compare. This one we have collected and observed. So that's why you need to draw the diagram of a sketch diagram of that microscopic diagram what you have observed and compare with this diagram rhizopus bread mold. Figure number 3. Okay, did you see this? Yes. Let's go for the continuation. So draw the rough sketch diagram in your notebook. Nothing but observation book. What you have observed. Take the help of the figures given in figure number 3. Just I have shown now like a bread mold, rhizopus, and compare. Okay. then you will get that. Now, observing bacteria. Observing bacteria. See here, observing bacteria. Up to now, we have observed that fungi. Now, let us go for the bacteria. Here, photograph of cardin bowl. This is suitable for that bacteria because in this lactobacillus bacteria is there. Okay. So up to now, we have observed fungi. So take a small, some rotten part of a vegetable or black spoiled part of bread or coconut with the help of a needle. Place it on a slide, put a drop of water, place a cover slip on it gently and observe it under microscope. And draw a rough sketch diagram in your notebook of what you have observed. Take the help of the figure given in the figure number 3, that bread mold rhizopus we have shown now. Like you just compare with your observation diagram. Okay, now observing bacteria, this diagram photograph of a cardin bowl, this is helpful for that activity, for observation bacteria. Okay, photograph of cardin bowl. So in curd, lactobacillus bacteria is there. Look, let us observe now about bacteria. So we can see the bacteria in buttermilk or curd or early morning scraping of tongue. Okay, scraping of your tongue with the tongue cleaner before washing the mouth. After washing mouth, nothing will be. We can be remove all. So before washing the mouth, you have to scrape the tongue before washing the mouth. We can also find them in the soil also, over bark of trees, over our skin, in our armpits, and many other places, but they are not visible to the naked eye because they are very micro. They are microparticles. They are microorganisms. So we can't see with our naked eye. That's why they came under the microorganisms. Okay, we cannot see with our naked eye. Now let us look at them more closely by the following activity. So here, this is a very interesting activity. See, look at here, figure number 7b, stained lactobacillus bacterium. Here, stained word here I used. So about activity 3, let us discuss in next class. But before that, here stained word is there. Stained. Already I told you, once again I will repeat. A dye, a color. Okay, a dye used you or here this particular shampoo, so as to make the structure visible for the microscopic examination. See here, we have used the dye. That's why a light blue color in that black aids we are observing. So to observe, to make the structure highlight, to make the structure visible for microscopic examination, we can use this staining. Very, very important. Okay, so here which is stained, that clearly will give that stained lactobacillus bacterium, or stained, like that they will give. Okay, so let us observe about activity 3 in next class. And so today we have discussed about the world of microorganisms and the great person Antonio van Leeuwenhoek and his microscope prepared by Antonio van Leeuwenhoek. And we had observed about, we had know that about microorganisms, and we had observed the different types of microorganisms like bacteria and shapes, about fungi and shapes, and protozoa and algae and microarthropods. In this, we may have chance to get bread mold, amoeba, paramecium, and chlamydomonas are very important for drying purpose. And a group of microorganisms and microorganisms in water we have observed, and we have observed fungi also. Okay, by taking bread mold, etc. And now we are going to observe the bacteria, lactobacillus bacteria. Okay, so as time up, I'm going to wind up the session. And thank you, Varandal. And let us observe a beautiful slides in next class. Thank you, Varandal.